Hey everyone, welcome back to the show. Today's guest is going to be Mark Hallen, and what Mark's going to be talking about is the process of molecular drug discovery using optimization algorithms. And the reason that I wanted to have Mark on is because, as you have picked up at this point, one of the main themes that I glom onto a lot of the time is the setting of priorities in your data science work. So, for example, prioritizing algorithmic needs versus what the subject matter and the domain requirements are. And what I find interesting about Mark is that effectively he has multiple needs that he needs to prioritize. And I think this is what really sets people out as a good scientific thinker, because you don't get everything. Not everything is very clean when you're trying to do scientific modeling and come to scientific discoveries. And what Mark is balancing is the an algorithmic issue, the complexity of the algorithms that it needs to use, and also the complexity of the different scientific phenomena. So basically, his work encompasses both the basics of physical knowledge with also basic chemical knowledge, and then the fundamentals of those topics, but then also the challenge that those physical and chemical fundamentals don't provide nice guarantees when you're working with, for example, large molecules. So it's the problem becomes more complex. Mark's problem becomes more complex as well. Anyway, so obviously rambling a little bit there. I've had a very productive week work-wise, but it does mean that I'm going into this one a little bit tired. So have mercy on me. Fortunately, Mark is a great guy, and I'm sure that he will carry the interview. So enjoy. Mark, welcome to the show. One of the reasons...
Thanks.
Yeah, I like to have early career people and entrepreneurs on the show, because usually they're about as sleep deprived as I am. So then we can really get that sort of grainy, edgy, brain melting down, edge of science type of feel to things. But it looks like you're actually a bit more bright-eyed than I am at the moment. So, just to get people real caught up on this, the things I find two things that are interesting about your work, even though I don't actually work in your field or very much approximately to it. But what stood out about your work was effectively I saw that you were having multiple prioritization tasks. And that's one of the things I think really helps define a grade A scientist, is that when they are essentially having to prioritize and do trade-offs in their analysis. And sort of the trade-offs that I saw for your work were, for example, the discrete versus continuous optimization aspect. And I guess obviously quite related to that is it's not actually two trade-offs for the actual mechanisms that you're studying, but I guess maybe even three trade-offs. So, you have the underlying physical principles, and then you have, I guess, the very basic chemical, like basic molecular understanding. But then the question is, okay, now you have a whole bunch, and what happens next? And so effectively you have these multiple trade-offs, and obviously your technical work is quite intertwined with the actual underlying physical principles that you're trying to capture. So anyway, that's what I thought was cool. And even if people aren't, directly working this field, it always helps to see a great scientist thinking their way through the problem. So that's where we begin.
Thanks so much, Glenn, for having me. It's an honor to be on the podcast and to get to share this with your audience. And I guess what I want to talk about today is an algorithm that I came out with in grad school and worked on in collaboration with my advisor, Bruce Donald, and lab mate, Jonathan Joe. It's an algorithm for molecular design, trying to explore really large chemical spaces, I think particularly for therapeutic uses. And it's an algorithm that we're now trying to apply at our startup, among other methods, in order to find some potentially promising new therapeutic candidates. So it's called LUTE, or local unpruned tuple expansion. And maybe I can start with talking about kind of defining the problem that we're trying to solve here. So I think it's well established now as a paradigm for drug discovery that you're going to try to find compounds that interact with some type of protein in the body, and generally that modulate its function, try to limit some type of disease process. Now, there are some pretty good techniques in this, and they work pretty well on, maybe 20% of proteins that are involved in disease. I think we've drugged something like that number. I mean, among the ones that are involved in disease, not in the whole human proteome. But we've largely done this through more either trial and error or sort of rational experimentation-based methods. And here, we search a large chemical space. You can even purchase something like 100 million different compounds. But that's really just scratching the surface of the total number of compounds out there. And one of my beliefs, and one that 1063 is, and really, the Donald Lab in general, have been, trying to pursue, is the idea you can target more diseases and you can modulate a lot more protein targets if you can expand the chemical space that you're searching. And, the possibilities here are almost endless. There's something like 10 to the 60, small molecules like, aspirin, penicillin, and so on, up to the level that sort of behaves like them in the sense of, drug-like properties. And here's just some exploration that people have done in the last decade of what they may look like. Some of these are probably pretty hard to make, but still, I mean, A2, you could maybe start to think about how people would make it. And then, when you consider proteins, when you consider drugs that are not traditional small molecules, but, that are kind of proteins themselves, and which actually have been a lot of the most impactful drugs recently, like, Keytruda for immuno-oncology. The space is even more endless. So kind of how did you explore this? Exploring a number of possible candidates that is, bigger than you can experiment on, and really bigger than you could ever hope to examine one by one, even on the computer. Sort of how you might start to go about that is going to be our focus here. So if you can do these bigger searches and, figure out the molecules that will best, interact with and block the protein you want to block or inhibit, I mean, there's the potential for impact on many diseases. And, this general approach, even with smaller chemical spaces, has already been tremendously impactful and behind a lot of so many of our advances in medicine recently. In cancer, which is, where this approach perhaps is used most of all, the goal would be specifically inhibiting proteins that either are mutated to cause or contribute to the cancer, or maybe they're just localized to the tumor or upregulated in it, somehow involved in the process. One project we worked on a little bit, it is in the academic context, so it's kind of what this is better with this. There's this protein CXCL12. It's localized heavily, although not mutated itself, in a lot of pancreatic tumors involved in the immune regulation, and being able to block that, kind of blocking the blue interactor here that naturally is there. Maybe even starting out from that, is one way that you could interfere with that process and hopefully start to be able to target these very difficult tumors. Anyway, just one example, because, if you look at infectious diseases, the same type of approach applies as well. You have these a lot of proteins that are specific to bacteria or whatever infectious agent you may have. I think there's a great need to develop new antibiotics because of the old ones gaining resistance mutations in those proteins that are targeted. So that's a very important area also.
Yeah, we need new antibiotics to fight all those new viruses that we misapplied them to.
I know, right? Well, the same principle applies to viruses as well. But of course, it's a different agent that you would want there. Yeah, I mean, I think that, it goes on and on. Even with Alzheimer's, biology, I think, is less understood than the others, but there's, some indication that the protein aggregates you see there might, they may be contributing to the disease. Maybe blocking that aggregation is important. And if not, there's probably other proteins involved in the disease process. So kind of a general principle here. Okay, so how are we going to do this? Like, how are we going to even conceptualize this bigger chemical space? Well, I think the one really tractable way to do it, and that does let you look at a lot of options, and this is kind of what nature does when you have proteins, and then, like, how do you have this huge space of proteins that biology has access to? It comes down to having a molecule with n components, where n is basically a design parameter. Basically n components that are mutable, where you can change the chemistry. So the classic example of this is a protein. So say we're doing protein design. If you look here along the backbone of the protein, this is always a regular pattern. You got this NCC, NCC, etc. Backbone repeat. Each repeat is called a residue. But what's different is on the middle carbon, the alpha carbon, as we call it, there's a different side chain coming off. So then you have these different chemistries for the different residues, which we call the amino acid types. So, this first one in green, that's valine. You have this 3-carbon side chain. For glycine, it's just a hydrogen. Usually we don't show the hydrogens explicitly. So that's why it looks like there's just a backbone. And of course, I think its goal is largely when you don't need specific side chain interactions. And then so on and so forth. So now we can start to relate chemistry to something that a computer can easily conceptualize. So we can construct this vector of the amino acid types. There's 20 natural amino acids for each. These are the ones that are going to be easiest to make, that biology across species can generally make. So we can have this vector, valine, glycine, serine, alanine, specifying the chemistry of just this short chain. And, they're going to be a lot longer in most applications. And you have this exponential growth of the number of options with respect to n, which quickly grows to numbers that are, well beyond, for example, the number of purchasable molecules or other, relevant limits there. Now, what are we going to do with this? I mean, obviously, we're trying to optimize R for some function. In some cases, you can fairly easily estimate a function, E of R, that you want to minimize E of R. Like if you want to stabilize a protein, E is really just the energy. Sort of the folding energy, I guess, is what you'd be going for there. And there's some, I would say at least qualitatively correct methods to try to estimate that in terms of local interactions. So that's kind of our picture here. Now, there's one wrinkle that I think I can expand on a bit. And it has to do with the fact that, to estimate energy, really to estimate a lot of these properties, you need to know something about the 3-dimensional structure. So so I have a backbone, generally for a protein, and in some cases for a small molecule, this is sometimes has additional complications, but sometimes it's useful to do this model. The backbone may not move that much. Kind of rearranging the backbone of a protein is a whole complicated deal with different folds. So there's actually the vast majority of mutations that would happen would not change the backbone that much. And it can be very, the most effective design probably involves taking advantage of a backbone that you already think is going to be useful. So we have the backbone, and then we have the side chains. Now, there's a lot in chemistry that is not that flexible, like bond lengths between two atoms with the same chemistry. That's going to be pretty fixed, but the bonds can rotate, so they're not going to rotate freely. You see here with the pentagons, that's the amino acid histidine, and you could kind of rotate around this first bond here. And certain conformations are preferred, so if you got a nitrogen up here, I think, for example, it may be advantageous to be at a 180-degree angle from it, like this one here, or there's a couple angles that are favored. These are rough numbers. It could be a little less, a little more. But the simplest model is just to say, okay, these are the canonical conformations. Maybe there's 3 for the histidine. Actually, there'd be a little bit more because there's other bonds that can rotate. But anyway, there's kind of the 3 main rotations, and then you can have these other chemistries. So, that'll get you to a simple model of, what can the 3D structures be. And just one important note, one of the reasons we can kind of fix the backbone is the sort of global rotational and translational motion is not really relevant to this type of calculation. It figures into some important properties like binding, but it's not something you're ever going to need explicitly model because you don't really care in the cell if a protein is upside down or whatever. So, that kind of takes some degrees of freedom out and lets us focus on the local flexibility that actually is going to drive our chemistry and our interactions. So what does this look like mathematically if you use this simple model? Well, we got our vector R, and each one will assign a rotamer, so amino acid type, but also one of these, discrete conformations, one of these discrete 3D geometries with certain, dihedral angles, which measures the bond rotation. So, maybe you could have histidine 0, histidine 1, these different, here with a certain fixed backbone, you can see what they are. So the typical notation here is something like, you got your residues, I, J, there's n of these. And then each one you can look up what rotamer, your vector R is assigned to it. So maybe I sub R could be, histidine 0, valine 1, the chemistry. And then also, what is the kind of conformational bin that's going to be in. And in our simple model, we're assuming like particular coordinates, particular, atomic coordinates in 3D for every atom once you assign the rotamer. So that gets us the flexibility aspect. And how are we going to model the energy? Well, the simplest models, they account for things like, for example, charge-charge interactions. So how would you calculate that? Well, I mean, every atom has, the electrons might be further or closer to it. So there's going to be a bit of positive charge or a bit of negative charge on most atoms. And then, in the simplest model, you would use Coulomb's law and just estimate, what is the interaction. And that adds up over atom pairs. You just add up, overall the opposite charge pairs, how much they attract over all the same charge ones, how much they repel each other. And then there's other also atom pairwise things like, van der Waals interactions. It's called, like if they get too close, then that's a big problem, as I'll get to in a bit. Anyway, what this leads to is, once you have a certain rotamer and a certain residue, for any two of those, you can compute this pairwise energy. So you end up with an approximation to the energy where every term depends on at most two different rotamer assignments to different residues. And then you want to, let's say you're trying to minimize E of R, and everything would just kind of a variant on this, but this is the simplest one. Then this is what you're trying to solve here, this equation here, when, E of IR and JR can be fairly easily estimated given any two rotamers. You don't need the full R for it.
Just out of curiosity on that last slide, since I know basically very little on the basics of chemistry, is there a sort of maximal plausible energy for any of these things? So the idea is, like, if you solve this and find that in trying to minimize the energy, that it's higher than a certain level, does that actually like preclude it from physiological possibility, or is oh, yes?
Yeah.
Okay.
Oh, yeah. That's actually a great question. So some of the most powerful techniques that we have are, taking advantage of that principle. As I'll show you, I think some of the models that I'm going to build here kind of separately look for these type of excessively repelling interactions. And then, find ways to take those to say, okay, we don't need to estimate this quantitatively. So whenever you're doing some learning on it, being able to eliminate those turns out to be critical. I will say, like, your models will, they basically go to infinity. If you force two atoms closer and closer together, you just get this enormous repulsion. They will not go to negative infinity. Like, I don't think, for example, you can really get a better, like a more negative interaction energy than, a couple of, +1 and, like -1 charges that are right next to each other. Like, I don't think there's really a chemically possible way to have, a +100 charge on an atom or anything like that.
Cool.
But,
and so is it sort of when you talk about the energies going to infinity, is the idea that these are actually effectively fairly well regularized functions because they will essentially react in that way fairly rapidly. So effectively, you aren't gonna really you can only, for example, push them together so much over you're immediately getting that super high signal, and your optimizer isn't going in that direction anyway. Okay, cool.
Yeah, exactly. Because you could theoretically do it, but in real life, a protein will avoid those conformations. The one tricky thing about this type of model is, since we are kind of fixing the backbone, we're saying, okay, given my backbone, I just want to see where all these pairs go. So you will be able to. There will be I sub Rs and J sub Rs usually, where the energy is just enormous because they're sitting on top of each other. But it's never going to happen. Like, the protein would unfold and the backbone would kind of blow up before you actually have that. So that is more of a model specific than a real reality. And if you know that your backbone cannot kind of have any reasonable energies, that's a good result too, right? Because that means, okay, this is not going to work. So
yeah,
you
cool.
need to know that.
Yeah. No, my immediate reaction when I saw this is like, it's like, I know you're thinking about in the physical sense, but man, like I've just blown up some optimizers in my days. Like Neville Longbottom blowing up the leaf, great. Just if there's an optimizer, I can effectively destroy it in most ways. But yeah, that is cool and helpful to get an idea about exactly how the actual function is operating. So thanks.
Thanks. Yeah. Yeah. Formally, that blow up definitely happens, and there's some interesting artifacts about just that phenomenon that have long played the field. But yeah, maybe that's a story for another day.
Actually, if you don't mind me asking real quickly, because obviously you're here and you're trapped in the screen, is there have you actually made any interesting discoveries when the optimizer produced unexpected results, or are you going to get to that? Like, have you ever actually produced some interesting new like intuitions about these molecular interactions by the optimizer doing something unexpected?
I won't say anything sort of physical has come of it. I will say there's an interesting phenomenon that has arisen about the models, which is something called the polarization catastrophe. So this actually, it doesn't come up with these super simple models like this here with the pairwise energies, but sometimes people look into, well, actually there's sort of a simpler version of it that comes up. Yeah, okay. I can sort of tell you about that. So sometimes people have kind of fit these models in different ways, and there's different types of atoms, and you assign charges differently and do van der Waals differently. So some people have found that in general the best estimates come from when you don't have van der Waals interactions for the hydrogens, so you don't worry about hydrogens getting too close to each other. And others in the field, including to some degree myself, think this is dubious, but if you do that, but sometimes you get I think the biggest issue is you don't have a van der Waals interaction, but you do have a charge at an atom. And as you know, Coulomb's law, as you get the positive and negative charges right next to each other, you actually can go to negative infinity, and that's totally not physical here because an atomic charge is spread out. It's not all at one point, but you model it in these models typically as being at one point. So anyway, this causes, I think, this is something they can really blow up the minimizer. There's sort of no van der Waals on a certain atom, so it lets it get close to others, and then the charges get right on top of each other, and just very silly stuff that can occur. So that's just one type of caution that needs to be used with these simple models. Yeah, that's probably
very fun. Yep. Yeah. All right. Yep. And now we'll go on to the next slide. But that is fun. Thanks.
Cool. Thanks. Yeah. So this model, I think once you've done all these energy calculations, which is, I think I've gotten to some of the messes here, but I think in a simple model, it's a little simpler because you know the coordinates, you just calculate the energies for them, and then you get something that is discrete enough that a lot of the sort of traditional computer science optimization techniques, actually at this point, you don't necessarily need to even get the sort of continuous minimizers like gradient descent or anything, and you can tackle it with these tree searches. The problem, okay, technically it's NP hard, like the way I have it here. You could think of E values where the cost would, at least to our knowledge, have to go up exponentially. But and generally, I think this is tractable for useful size systems. You're at least for the kind of stuff that we usually do, we're looking at an active site of a protein. We're looking at maybe the interaction between two proteins. There's not going to be like 1,000 residues here. So it can be tackled with a couple different types of techniques. What generally works best is kind of, you have these nodes where you have a vector R with, that's sort of not fully defined, sort of a partial conformation. Like if R is a conformation, it's the chemistry, and 3 coordinates of all the, of all the different residues, partial conformation tells you for some of the residues, and then the rest remains unknown. So at your root node, you start, you don't know anything about the osmolar R. And then maybe you can split that node and you get all the possible options, all the possible rotamers. If you allow all 20 amino acids, which you don't always want to do, but if you do, then probably you got, a couple hundred rotamers. And so you get all these, child nodes, and then you can split that. And then, the algorithm will tell you which nodes to split and to expand, the next residue down the line. until you got them all. And these techniques, there's both kind of depth first and best first techniques like DEA star that can get you the optimal. They can get you the optimal list of rotamers, the best R, and they can even get you a gap-free list of the best, second best, third best, and so on. These models are approximate, so having multiple to test out experimentally is always helpful, I would say. At this point, you're not going to be certain. And what algorithm you use, the LUTE is not kind of focusing on this area, so I won't get into it too much. But largely depth-first approaches are good if you're trying to save memory, which is often important. Whereas, A-star-based approaches can, under some circumstances, guarantee that you are doing the minimal amount of node expansions that you need.
So, just real quick, because I know that some people in our field aren't as familiar in the optimization arena. Although we all should be, because as long as we're fitting maximum likelihood estimate models, we are doing some optimization on a daily basis. But on this one, when we talk about the depth first search, so the idea is that effectively we are going down as far as we can into the graph. And it's once we've explored all those areas, then we start working back up and start checking the other, the combinatorial elements. And I was going to relate this to greedy algorithms in some way, but it's actually I had an analogy and now it's popping out of my head. The equation that you showed in the previous slide actually is, as you said, technically NP hard. However, on a practical sense, and some of that is because effectively we have these, I guess, I wouldn't say them impossible, but these like implausible regions against which the optimizer would not be forcing the search. So essentially, just to use the term lightly, the physics is forcing the search into a certain direction, which then makes it transitions from being technically NP hard to now effectively being a much more practical, tractable search. And so then, you said that there are some. So are these guaranteed to have a local minimum, or are you? Are you guaranteed to achieve a global minimum in these?
You are, yeah. So the algorithms that I described here, they're guaranteed to find the global minimum. They are not guaranteed, any tractable level of complexity. So, they could take exponential time. And to be honest, as n gets very big, they do become quite intractable. There are problems that some people try to tackle, like trying to design all the residues on a protein. Sometimes they finish quickly. Other times they just blow up and you get nothing. But yeah, you will get the global minimum. You can also go the other way, which is like, okay, I have a certain amount of time. I'm going to use, simulated annealing or something. And in my estimation, I would say if you're trying to do a whole protein, a lot of times trying to stabilize the fold, you think there's a lot of answers that'll work well. People have gotten pretty good results with, simulated annealing type techniques. So like, basically they want it to work reliably for very large n, but they don't really need it to be truly optimal. Whereas I think if you have a small number of residues, you really need to get the chemistry right in an active site, like to make some kind of therapeutically relevant interaction. Then maybe, your n is probably not that hard. You probably can really get the global minimum. And it's maybe more important to do it. So then these types of techniques may be more effective. And we've seen some kind of successes toward that direction, I think, using them.
Just for some fool's intuition on this, so I guess the way that I was doing it is effectively for these smaller molecules, effectively you would want to you work out one area first, and that is the depth first search. And the reason it makes sense is because obviously, in one regard, whatever little bit you're working out is actually a quite large portion of the total phenomena, the total molecule that's of interest to you. And when you said that, effectively, when people are looking at larger and larger molecules, is the idea that, and you can do some of these more like breadth based optimizations, that effectively there's enough going on in the molecule that you don't need to get it perfect, and that there's effectively there might be some larger interaction, larger physical phenomena going on there that doesn't actually require you to actually know certain specifics while still getting it generally correct.
Yeah, basically, maybe I can actually several slides back. I have a picture. See here, say you have this protein, you can imagine doing a couple things here, right? So first of all, you can imagine, okay, let's say you're trying to, I'm not sure you would want to do this, but let's say you're trying to design the orange protein like this one. And then maybe you're trying to modulate its interaction with the blue one. So as you can see, there's only like, a smaller number of residues that are actually close enough to the blue protein to matter for that. So that would give you a smaller N. And those are kind of the sort of, activity related residues. And yeah, getting that precisely right is probably important. And so, whereas there's another problem you could do, which is like, okay, maybe I want to stabilize the whole protein, or maybe it's even like I just want a totally new protein that kind of has the same general fold, like the same backbone, but just different side chains. And for that kind of problem, yeah, that's probably if it's this requires a lot of biochemical intuition. You got to think, okay, is this a problem where lots of sequences will work? And then, yeah, it's large N, but large N is not a problem if you're using, simulated annealing or something else with sort of, kind of a fixed time cost, and then the accuracy will suffer if you can't have both, so.
Cool. Actually, another bad metaphor popped into my mind. That I was thinking, it's like these small molecules. It's a little bit like the Sudokus, where if you have a tiny Sudoku, you really need to get each of those squares correctly. But if it's big enough, no one's gonna mind if you have a few extra ones in one thing and things like that. But perhaps that is inept. But yeah, I'll let you go on.
Yeah, I think there's something to that. Yeah, there's more room for error. Even just a biologic in general, as compared to like a true small molecule, I think there's more room for things that are not quite ideal, just because the interaction surface. Even here, these are both considered like large molecules in drug world. They're like a small molecule is, usually under 500 Daltons, and any one of these residues could be 100. So, but yeah, good. That's actually a pretty apt point. Anyway, so this is a discrete problem solving this. Okay, now we kind of get into where LUTE can help us. The problem is basically, molecular flexibility is not really discrete. Atoms can move in 3D continuously, and not just sort of the whole molecule rotating and translating, but even just the bond rotations, all the relevant local stuff. So there, yeah, I think it's correct to say in most of your designs, the backbone is roughly fixed, and the side chain is roughly in these preferred conformations. But if it rotates, maybe 15 degrees either direction, the backbone tweaks like this. I mean, that's very much within the realm of possibility. There's not going to be a huge energetic cost to that. And so I think to really design properly, we gotta model the fact that it can do this. And then, moreover, the best models of energy are not going to be exactly pairwise either. I mean, the Coulomb interaction assumes points at the atoms. But they polarize each other. There's water everywhere. All that stuff also ideally should be modeled. You can think of the pairwise energy as just kind of the second order truncation of it. And to be honest, the models that are explicitly pairwise are probably not even that close to being the exact second order truncation either, because of all these other phenomena that are a little complicated to model. And in fact, what I'll talk about a little bit is getting something closer to the true second order truncation, which turns out to work a lot better. So yeah, in this case, the E of R for a voxel. Now your R is still a discrete variable. It's still a list of rotamers. But instead of just being the energy that you calculate from the sort of ideal 3D coordinates at each rotamer, instead of that, what you want your E of R to be is the minimized energy in sort of a voxel, like a sort of region of conformational space around each rotamer. So basically, out of all these conformations here, the blurry ones around the discrete rotamer, which is shown here, the minimal energy for that should be our true E of R. Anyway, I just wanted to show you, like, when can this be most important, the continuous flexibility. Like I said earlier, one of the most dramatic phenomena you see in these energies is when atoms that are not bonded to each other get really close together, which we show here in pink. And that just blows up the energy like crazy. And so, if you were just using your ideal rotamers, I mean, the sort of ideal conformations for each amino acid, I mean, when you model those, you don't know anything about where these different pieces of backbone are. So, you could easily just be jamming them into each other. And then, a 15-degree rotation one way or the other, even like, 5 degrees sometimes. Yeah, that could make the difference between clashing and like pretty bad energy and something that maybe is really what you want. So these are just small shifts here. So this is perhaps the most dramatic case of continuous flexibility being practically important, as the Don Lab has studied for many years, even before LUTE. And to account for that, they adapted the DEA star algorithm to include this type of continuous flexibility. However, the computational cost is rather steep. The basic idea here is that, you would have kind of a lower bound on the energy for each R that is pairwise decomposable. And then you would enumerate an order of that. So like I said, you can get a gap-free list. This generates a huge amount of conformations, kind of a bunch of these vectors R that you've got to do the minimized energy for. And that can grow exponentially with respect to n, both the number of minimizations and the memory cost within sort of the tree due to the nature of the algorithm. Unfortunately, this does limit the amount of chemistry we can search. Often we keep n rather small. So, of course, the root cause of this is really the factor graph blowing up. When you have that Coulomb model, when you have a discrete pairwise model, you have these energy terms that really just depend on the rotamer assignment to two residues. But once you're doing a locally minimized energy, an E of R for a full R, I mean, that depends on all the rotamer assignments. Bless you. That makes things more complicated and causes a dilemma, which is, either we can model these physically important phenomena like continuous flexibility, or we can use these efficient discrete search algorithms that assume, either that discrete pairwise model, in that mathematical formalism or something like it. So that kind of is where LUTE can come in. The idea of LUTE is that we're going to take, E of R's calculated with this complicated model and think of what is the problem of getting the actual optimal R accounting for all that, and then reduce it, at least with negligible error, into a discrete combinatorial optimization of the same mathematical form we had in the discrete pairwise model, just with, different numbers that reflect the continuous flexibility, that reflect, hopefully, some better energy functions, and then solve it with our efficient discrete algorithms. So it looks like this. Basically, we're going to try to learn an expansion similar to that. So what is E of R? E of R is that minimum energy, continuously minimized. And we're going to model it. We're going to try to learn a model of it as a sum of coefficients. And generally, these are pairwise things. I mean, I think the sum of M and T generally will be similar in form to that sum of single and pairwise terms. Sometimes we would include triples. But instead of just kind of being able to calculate these directly, we learn them from samples of E of R. And then, there's all these tuples in R, the pairs of rotamers are different residues and so on. And then, given R's that have all these features, then it's just really linear fitting. And so what that does for you is, you know how to solve this problem in A, sort of find the lowest energy red dot, but you really need to find the lowest energy blue curve, based on its lowest point on each. And then using this decomposition, you can kind of easily predict what is the red dot for each blue curve and solve it that way. Now, one important thing here, and this actually touches, Glenn, on something that you asked about. This expansion cannot actually be learned with great accuracy, at least with pairs and sparse triples, if you want to say, okay, I want a good protector for every R. But, that's because of all the R's where E of R is very large, and you're never going to want to have atoms on top of each other or anything like that. So we got to prune these. And there's also shown here, these are kind of from our first paper on LUTE. Since then, we've actually used, and this is based on energies. Since then, we've actually found some geometric techniques, kind of partial plug, pruning of low unreasonable geometries that, really kind of lets us explicitly get rid of the classes. But for the minute, I think we can talk about this fully energy-based framework. And what's going on here, if you can ahead of time remove the high energy components from the expansion, then that gives you the ability to get a more active expansion. And what I think is going on here is, the adjustments that you see in a LUTE expansion compared to a discrete pairwise model, like how are our pairwise energies, so we learn different pairwise energies that you compute without continuous minimization. It's really based on the shift that you see in a residue pair because of all the other ones around them as they locally minimize. And if there's no clashes, this will be subtle. I think you can kind of cut off the second order, sometimes third, pretty accurate. And then, I mean, that's all good. If there's a clash, now you're introduced to boom, they're so close to each other. Everything else will just scurrying out of the way to adapt for that. And I mean, that makes it hard to fit. So that's probably what's going on here. Given that, you can use some pairs and some cross triples as your features. If your model looks like this, some of the tuples you see in R, the coefficients for them, and of course, if one of the coefficients you've identified as bad is there, then you just try that quantitatively estimate the energy, and you can exclude that from your search. And so that leads us to pretty accurate energies. So even just the source implementation, we found that generally the difference between the actual local minimized E of R and the estimated E of R that we learned with our lower order expansion, our local unpruned tuple expansion. That's expansion over unpruned tuples. So with this error, the mean square error is typically below any level that would be biologically significant. So a pretty good estimate of that is sort of what's the thermal energy or room temperature. Fluctuations in energy you see just kind of randomly with significant probability. And that would be about unit is kcal per mole squared, so it's about 0.36 in this level. So most of this here is well below anything that would make any difference. As we zoom in, the couple of examples are worst. I didn't include sparse drills, so it was just pairwise expansion. I got that in blue. There was only about 4 points with an energy that I would consider biologically significant. And then for 3 of them, when I added some sparse triples, it then kind of went down to where it's pretty benign. There's one where it was still some, problematic error. I looked into it and I realized that I think what was going on there is our local minimizer was not actually finding the right minimum within a voxel, So like we have these side chains that are supposed to, in this model, they were supposed to go up to 9 degrees in either direction compared to the ideal one, and find the minimum energy within that range. But it wasn't really reliably doing that. So that's a whole other set of issues, I guess. I think plug has helped with that. But this is what we had kind of, from the improvements from LUTE by itself. And what that gave us, it roughly doubled the amount, basically doubled the end that we could handle compared to the immediately previous model, to be able to model all these residues with continuous flexibility, and expand a lot less nodes than DEA star. So that sort of speeds up the tree search compared to the bounding based techniques by 2-3 orders of magnitude. And moreover, it also helped a lot, not just in sort of minimizing E of R, like stability type designs, but also more sophisticated designs, which are often of much greater significance in actual therapeutic related applications. Actually, whether you're designing a protein or a small molecule where you also have these mutable units. And a lot of times you're trying to be selective, for one, you want your molecule to bind one target, interfere with it, and not another. Like there's a couple of proteins in the body. Simplest example, maybe there's like a human and a bacterial protein, and you only want to go for the bacterial one. Anyway, in sort of some example cases of this type of model, what we saw is that the complexity of using LUTE, using this continuous to discrete expansion, although it would give you the same results as sort of the results in green, where you do an exhaustive search over the molecules, which was previously the state of the art, at least for any guarantee of accuracy. Basically, you saw that the complexity of doing this with LUTE is far more similar to the complexity of using a discrete search than it is to the previous continuous methods. So it kind of gives you the benefits of the continuous method without most of the design computational cost that comes with it. And we also had some success in fitting to models that are not intrinsically pairwise. Those examples I've shown you before, just because the pairwise energy models are pretty fast to evaluate. We mostly did our continuous minimizations with them. but we're trying to build up to where we're using more advanced energy models. Like this is basically energy when you know the 3D coordinates. So whether it be from a continuous or discrete model where you know where every atom is, you want to estimate sort of the interactions with water and stuff. So we got good residuals too. All right, so yeah, that's LUTE. Basically, it's a discrete expansion. What it does is it's meant to get the same results as if you did a design, fully modeling, continuous flexibility of the proteins. Let them move continuously in 3D, and or the small molecules, like whatever. If you're designing a therapeutic, whatever it is, how it interacts with the protein it's trying to block. And then sort of these energy functions that are not just sort of based on atom pairs. And then we want to do all that with a computational cost similar to those discrete pairwise models that have long been prevalent in the field and that many people have studied and found very fast ways to handle. And so what this lets us do is to bring in more accurate models as we attempt to identify promising therapeutic candidates from very large chemical spaces and kind of leverage this ability to change the chemistry at many positions in a molecule to see which might be best for whatever impact we're trying to make. So yeah, and then I should probably definitely mention and thank my collaborators on this work, Bruce and JJ, and of course everybody else in Donald Lab where I did it, and the Pharma and Lieberman Foundations and National Institutes of Health for funding.
I want to ask just a few comments or questions while I try to dissect that. And then there's actually a side conversation that you and I had, obviously, that we had started prior to this about basically just actually where cool machine learning techniques can come in with regard to like how much data you have and where, for example, like maybe a direct simulation versus essentially a more tangential, that's not quite the right word, but a more like abstract approach would, and that's not the right word. I'd rather go for that tangential approach, where effectively whether or not you would want to be deriving things directly from a simulation based on your knowledge of the physics with underlying principles versus simply trying to learn use the data to learn the function that sort of maps these relationships. But very first, so I guess I thought it was interesting that effectively the little components that you are trying to optimize in there, you have each of those pairwise interactions, but of course that is that's not quite reality. It's just sort of essentially a good estimate of reality, because obviously the correct one would be like all interactions within some sort of like physical bubble or physical limit, presumably, because that would be the true nature of it. But the fact is, many of those would be either zero or infinitesimally small. And so is that correct? Yeah.
Yeah, that's true. I mean, I guess what you would really want to do is, not use Coulomb's law at all. Well, okay. I mean, you wouldn't use it in its classical form, at least. You would have quantum mechanics. As for going to zero, that does happen. I mean, I think like the most tractable quantum mechanical models may also take advantage of the sparsity. Not all atoms are really interacting. I mean, the ability to do that practically, it comes to varying degrees. But yeah, I mean, I think what you said is right on.
Cool. Yeah, and so I guess was there? I guess one of my things is I'm always curious about what is the underlying mechanism that people actually appreciate in there. Is there and perhaps you've already answered this, and I'm just not familiar enough with the physical terminology, but like is the beyond just sort of the computational need, is there a practical need where effectively from the nature of these molecules that effectively you don't even need to worry about them? And also, I guess I was also wondering, is there any sort of categorical, isn't the right word, but discretized element to the nature of the molecules themselves, such that you would actually not need to worry so much about any pairwise interactions, or you need to worry about them more?
Yeah. So basically, do you mean, is there a way to sparsify the pairwise interactions so that you don't need to do all of them? Yeah.
Yes. Yeah.
Oh, yeah. Yeah, that's actually a pretty interesting area of research that some people in the field have focused on. So JJ, who worked with me on this somewhat, focus a lot on that. Anyway, the field has found a couple of useful things. One of them is even not dealing with continuous flexibility. First of all, you get small. Even just based on Coulomb's law, without making any new assumptions, the farther that charges are from each other, the less they interact. And that can even be compounded, I think, under certain conditions when you have like dielectrics. It's really still linear, but I mean, some models that have, for reasons I don't quite understand, fit there even seem to find like a higher degree of fall off, under certain conditions. Anyway, what that leads to is that, it's really the residues close together that matter. And to be honest, beyond the Coulomb interactions, the van der Waals interactions actually are very important here, and they fall off, much faster. Like, the distance to 6 power is the dominant term there. So even if you take these models and calculate energy with them, and then you can kind of get a sparse interaction matrix based on either energy or distance cutoffs. And that tends to have a lot less elements in it. So that's one advantage. But it turns out that the NP hardness under some circumstances can go away. Like if you can draw an interaction graph between the residues, like which one have non-zero pairwise terms, you can calculate what they call like the branch width of the graph. It's a sort of a quantity that, it will be n for a dense graph, but it will be like 1 for a chain and then kind of go up in between. And then the complexity, I think, forgetting the exact exponent, but I think it goes up as something like the 1.5 power. What was it? It's exponential in something like 1.5 times the branch width or something like that. It's not exponential in n the way that the dense graph is. So anyway, those results I think were very cool. And then of course in a LUTE context, when you're doing fitting, one thing we mess with a little bit, but haven't actually kind of yet put into our models for full production is, regardless of the branch width, the less terms you're trying to learn, the less samples you need, so you can kind of expedite your learning process and everything. So that's all definitely useful.
Yeah, one of the just to sort of hop a little bit, one of the things that sort of my mind wandered off on when you're talking about when people were trying to do these less depth first and more of a breadth first search was essentially the question of it's like, well, are there certain sort of effective dimensionalities on which you can effectively save yourself time if you can identify those? And because I figure it's like, well, I bet there's some physical principles that would allow you to rapidly do that. And I wasn't sure if there's any ones that you would actually just sort of discover from the data or experimentally. Because that, I guess, to me, I was a bit fixated. It's like, well, I wonder if that could actually be just derived from the basic principles of chemistry. So the effective dimensionality aspect was something that was of interest that I was thinking about quite a bit.
Yeah. So I think the branch width gives you some of that. So that sort of maybe effectively reduces n to some degree. The other thing I think is, so you can definitely, so you're talking about sort of something to the n, right? So like there's a number of options in every position, and that's something you can really cut down. So yeah, first of all, as you mentioned, a lot of rotamers clash with each other, so that's not going to work. I think that in another thing is a lot of times, due to your lack of confidence. Okay, I don't know if you use such strong words, but okay, due to the intrinsic uncertainty in basically any models used in computational chemistry today, it often can be advantageous not to be too aggressive about changes. So, for example, you often may want to preferentially explore mutations in a protein that are amino acids with similar chemical properties. So, for example, in these coronavirus mutants that you see recently, you had this very dramatic, strain that they first noticed in South Africa, and it's like a mutation that flips the charge. I think it was from negative to positive at a certain amino acid, and those are going to be harder to predict. And also, it's probably less likely that they'll work. I mean, sometimes they do, but in a sense, the coronavirus was trying to make it very different so that it evades its target. So unless you're trying to do something like that, you may actually significantly reduce the dimensionality by saying, okay, I started with a negatively charged residue here. I'm going to keep a negative charge, and so on. So that's one advantage. And, the other thing is, I talked about proteins a lot here, but, a lot of the very interesting applications of this type of technology, and certainly at 1063 where we've been focusing actually, is in designing small molecules. And here there's this whole nother set of constraints, right? So like, first of all, small molecule is smaller, the N will be much smaller. The number of options is often much bigger because you're not just 20 amino acids, you're all of chemistry. But then, you're often going to want to, a small molecule, each one has to be synthesized differently. That's harder than proteins. So the considerations of which side change can easily be put in from a synthetic perspective will be a very large dimensionality reduction in that case, as well other pharmacological properties. Like to the extent that you can predict what will get in the body, what will be sufficiently soluble in water. Those things can also help you to reduce dimensionality when also improving your your actual drug likeness of the molecule you're making.
Yeah, just real quickly, and this will rewind back to one of the things you're saying when you said obviously some of the rumors are impossible because they'll effectively crash into each other. Is there a way that you can actually algorithmically like pluck that out, or is it just bad luck? Like, is there or for example, can you put in an addition, or is there an additional, like, is one of the sort of part of that cost function the crashiness, or is it all would already be encompassed by the energy measurement to begin with? I guess that would be it.
Yeah. So the energy does do it, but actually that is something that the idea of explicitly looking for that, it is something that has been useful. So so the energy, you can prune based on energy. Like if in D A star, it's actually two phases. It's a pre-pruning based on energy, and then the real thing. So in the kind of the initial loop that I showed you here, what we usually would do is we compute in pairwise energy. So this requires trusting a pairwise model, at least at that stage, some type of pairwise model. And then you prune based on that. But, I think as you go to better energy functions, ideally you would want to work exclusively from E of R. You would not want to assume any type of pairwise energy. But what you can do there instead, and actually I found this to work better. I found this to give me better fits in many cases, and to resolve some of the need for triples and stuff. I found that you can kind of calculate based on geometry. Are these atoms going to clash or not? Because to be honest, these energies, every interaction here is qualitatively correct. Opposite charges will attract, same will repel each other. You can't get too close. That's all true. But, and we can estimate it, right signs, probably even for residues most of the time. But, what I think we know with more precision is the local stuff, how close can atoms get to each other? There's all this work been done on molecules, looking at their 3D structure using X-ray methods and stuff. And so then if you prune based on that, and then you use some geometry to say, okay, given the continuous flexibility, how close could these atoms, how is it possible to maneuver these rotamers so they don't get too close? Those types of calculations, plug is the name that I have for that algorithm, which was kind of a next step after LUTE. That I think directly touches on the question of, what are the bad pairs and sometimes triples that you want to avoid. And then you can sort of have the true, learn only from E of R method. And you don't need to, rely on the energy, on sort of looking at the energy to get that thing. So
cool. And so is it, I guess, is it more like if you had the entire search space and you've now Swiss cheesed it, as in like there are bits that are missing that you don't have to, or is it more like you've actually done the branch and cut? Is it more like a Swiss cheese thing because of the continuous nature?
Yeah, I think of it more as like Swiss cheese because, so imagine you're expanding the tree. Well, okay, actually, to be honest, it's both. Because when you're looking at clashes, sometimes you see, oh, this rotamer will never happen. Like there's literally no set of rotamers I can put elsewhere that would work. Like for example, if you have a rotamer and it clashes with like the backbone, and the backbone is not moving enough to make it fit, then that directly reduces the dimensionalities, of your tree. It reduces the number of branches. Whereas, if you have a pair removed, I mean, you can't say until you expand the tree, where you're going to cut there. I guess, I guess, once you've branched on one of them, then branching the other is limited. So I guess it helps you there, but it definitely is a little more like Swiss cheese in that sense. But it's something you can take advantage of during the tree search. It's just not sort of I wouldn't characterize it as always being just a matter of dimensionality reduction. It's something that you have to dynamically account for.
Cool. And I guess maybe if you have a little bit of time, maybe one of the practical interests, just for people who are interested in my Swiss cheese versus branch brown cut analogy, that what you're doing with your company. So, like, where are you actually going to be taking this and implementing it? Where do you start? Where's, for example, an area where you would immediately want to be applying it?
Yeah, thanks. So I think our biggest interest is figuring out how these types of techniques can help us to discover potential new cancer therapeutic candidates. So we're kind of focused on small molecule design because that's, I think, an area where there's several particular targets that are sort of within the cell, hard for anything but a small molecule to get in there, at least in large amounts. It's been shown biologically that blocking them is important. In one case, there's actually this, there's this mutant. There's these mutations that cause cancer. So there's this one protein, KRAS, in human cells, and it has a mutation. There's a certain residue, glycine 12, changes to the amino acid cysteine, and cysteine is a little special. It turns out that it's fairly tractable to get drugs to make chemical bonds to it, and that gives you a very selective type of drug. Anyway, a couple of companies like Amgen and Mirati have found drugs that do this, and using in some cases, well, I mean, they haven't revealed all how they did it, but certain advanced experimental techniques kind of pushing the limits of searching chemical space just experimentally. I think that's at least what Amgen did. They used this, I think, real cool technology from a company called Carmet, I think. But what they anyway, so they found this thing that you make a special chemical bond. And it's actually in the clinic, and it does seem to be helping, for certain cancers that were very intractable before. And I think even more excited to see what's going to happen when they combine it with certain other drugs, because it's, for a cancer therapy, it's remarkably low in side effects because it targets just a mutant. It targets a protein that a healthy cell just won't have. Now, but the problem is, most people who have cancers of this type, the mutation is not to cysteine. It's at the same residue, but a different amino acid type. And you can't make the covalent bond there. So that poses a very difficult, problem that has actually for decades now, chemists have been going after variants of it. And we think, for example, that if you could go and design computationally a small molecule to do that, so we can get into the cell and it could be selective. I mean, I think you're going to have to search a very large chemical space to do it, because smaller chemical spaces just weren't working. But if you can do it, I mean, it would probably bring a lot of the same benefits that, for example, Amgen's molecule, sotorasib, if I'm pronouncing it correctly, I'm not sure the stress is, it would probably bring a lot of the same clinical benefits that sotorasib does, but to actually the larger population of patients who have other mutations that just happen not to have this kind of easy chemical bonding strategy available. So that's one of the problems that we are very interested in.
Yeah, on the issue of sort of other mutations, how does tumor heterogeneity play into this? Because one of the things I was saying is, like, it's really great when you have certain things like KRAS, where you essentially have this lighthouse signal that you can go and you can aim for. One question is, I literally don't know the answer to this, so I'm not even leading you. But like on the surface area of the tumor, where your molecule might even what would be doing the interaction, is there any issue with the tumor heterogeneity where effectively there's more of a certain type in the surface, so the surface has one type, and then there's more of a mixed bag in the center? Or am I misimagining things, given that effectively, these things have access to the biocentration, and therefore there's ways that you can actually access the entirety of the tumor at once?
Yeah. You point to a couple of very, very thorny issues there that I think we are well, frankly, a lot of us in the field are trying to think about how to handle. Um, so yeah, there's a couple kinds of tumor heterogeneity I think that are very serious issues. One is sort of the structure, that the inside has there's sort of this very kind of hard to penetrate environment called the stroma in some tumors, especially in the pancreas, where KRAS is often a major mutation. So yeah, getting in there is important. I think a small molecule can get in more easily than a protein, but still, you may need additional treatments. I think, to be honest, for these difficult tumors, it's hard for me to imagine that a single drug is going to do it. I think it's to really get rid of one of these tumors. I think I have one that some of the approaches being developed now can do it, but it's probably going to take several. And what some of them is probably going to need to do is to kind of maybe not break down the heterogeneity, but at least make it easier for either immune cells or drugs to get into the middle of it. And then the other thing is the heterogeneity in terms of different mutations that occur. So like, I think that really the biggest thing that plagues these kind of sort of targeted, like protein targeted approaches to cancer, as opposed to, surgery or something, is people develop resistance. There's so many of these molecules, like you give it to someone and it works well for a while, but then there's always like well, not always, but there's often like a few cells that have some mutation in the protein of interest or in another protein kind of related to it, that doesn't respond in the same way, and maybe 6 months, a year down the line, now those are the only ones that survived, and you've got a tumor full of these resistant ones, and then you're kind of stuck. So I think there's a couple things that we have to do here to combat that, and one of them is see what mutants in other proteins are involved in resistance and target those. And then the other thing, frankly, one thing that the Donald Lab has worked on somewhat, and that I think would be very interesting if more can be made of it, is predicting these resistance mutations. Like if you see, here's a protein, here's a drug that's going to try to block that protein, how can the protein mutate that will block that drug but still be able to do the natural function? So actually our methods kind of related to, basically this continuous cycle approach was the first one to be used to predict ahead of time what such mutations would arise. And then that hopefully can help us to go back and design the drug. So that's something that I think we're interested in working in over time as well, is can we try to leverage this knowledge to design drugs that will overcome it. And that's difficult, but it may be necessary. And maybe it's one drug, or maybe it's a cocktail. That's what worked for HIV when they overcame some real nasty resistant mutations. So that's heterogeneous in all its own way. But yeah, so basically, yes, tumor heterogeneity is one of the biggest challenges here, and it's something we always have got to think about to overcome here.
Cool. Well, thanks so much for your time today. Is there anything else I did want to cover that conversation that we are having a bit earlier about basically the data issue, but I fear that honestly, maybe it'd be good just to set that up for a different, like set that one up proper. And so and bring in a few extra examples. So I guess if you're around, it'd be great to have you back on the show at some later time that we can talk about some more of these issues, because I think you did bring up some very good points on this other issue, and I thought it was very interesting because I saw some massive parallels in it between the work that I've seen for essentially personalized expectation for patient health and sort of digital phenotypes in that way. So yeah, I guess Mark Hallen will be coming back on the show sometime to regale us with more stories about blowing up optimizers and generally fun stuff like that. Hey guys, it's Glenn. Thanks for your time today. I hope you liked today's episode. If you did, please consider smashing that like button. It's the single, simplest, fastest way to make sure that YouTube shows this video to more people. If you really want to go crazy, consider subscribing or going to our website and joining the mail list. If you want to go totally crazy beyond that, forward this to a friend or colleague who you think might enjoy this too. We're a small channel and every bit helps. Our next episode will be coming out next week, so in the meantime, feel free to look around the channel and see other videos that might be of interest. As a quick disclaimer, the views expressed in the show do not represent anything other than the people saying those words, views, etc. like that. It doesn't mean anything about their employers or their employers' views or some thing about their employers or their neighbor's cat or anyone else not saying the words. And in fact, given that people tend to change their views when they're thoughtful enough, it might not even represent the views of the speaker by the time you're hearing the episode. So definitely come back and see if they've changed their views at a later date. They also don't represent the views of our sponsors. Thank you to our sponsors. You can check them out on our website.